Good morning, everyone. My name is Andrea Layedra. I am a CONICET doctoral fellow, and my doctoral research focuses on studies in C. elegans through the applications of microfluidic devices. The study that I'm presenting today is called a microfluidic device for simple quantifications of chemotaxis with Chemotaxis elegans in neurotoxicological studies. As a background, nowadays toxicology must meet two opposing demands. First, the analysis of a growing list of chemicals, including pesticides and insecticides, that are developed to many important purposes, like protection against crop loss and against vector-borne diseases. However, there are significant concerns over the potential toxic effects of pesticides to non-target organisms, including humans, which may even cause degenerative diseases such as Parkinson. And the other demand is the resource limitations and ethical concerns associated with traditional mammalian testing. In this sense, Chemotaxis elegans is a useful neurotoxicological model that has been successfully used in many investigations to study human neurodegenerative disorder. C. elegans is able to detect external chemical signals and perform chemotaxis behaviors through its simple chemosensory system. So here we propose that the neurotoxic effect of xenobiotics could be reflected in the chemotaxis behavior of this nematode. Here you can see the traditional method to perform chemotaxis assays. C. elegans chemotaxis is traditionally assessed on agar plates. In this image, you can see briefly a point source of a particular chemical is placed on an agar plate for establishing a concentration gradient of this compound. The nematodes are then placed on this agar plate and left to crawl according to this concentration gradient. This assay is analyzed by counting the number of nematodes in the source area of the chemical. However, this method suffers from several disadvantages for obtaining reliable data. First, the conventional methods are rather time-consuming due to the slow diffusions of chemicals in the agar plate. Second, the ability to create and maintain a desired shape of the chemoeffector gradient is critical for analyzing chemotaxis of this nematode. However, it is difficult to control the shape of concentration gradient in this method. And finally, only the number of C. elegans in the area containing the highest concentrations of chemoeffector is counted for analysis. The chemotaxis to other lower concentrations of chemoeffector is difficult to be simultaneously examined on one plate. It's for those reasons that new technologies such as lab-on-a-chip devices have been developed. These devices are used as analytical assistants for handling of organisms. Between the main advantages are the low cost of manufacturing, there are easy handling of nematodes in this case, and the nematodes are loading by suspension. Regarding the materials, this device can be made of different materials such as glass, silicone, polycarbonate, and polymethyl methacrylate and polydimethylsiloxane, or PDMS. Unlike other materials, PDMS is used for biomedical applications, cell culture, implants, among others, due to its properties. It's a silicon elastomer. It's biocompatible. This device has high oxygen permeability, is transparent and economical. Okay. With the aforementioned, the following objectives have been proposed. First, develop microfluidic devices for toxicology studies. Second one, determine the methodology for chemotaxis analysis using microfluidic devices. And finally, the applications of microfluidic device to toxicological assays. Regarding the first objective, the PDMS devices were developed. For this, the methodology is as follows. First, the design is elaborated using Corel program with the desired dimensions. This design is printed on a polymer that serves as a base for an epoxy resin mold. Then, this resin mold is filled with PDMS, where more replicates can be generated. The PDMS device is bonded to a glass slide using a plasma, and that's it, the device. In this figure, you can see the designs we are working with. This is the one I talk about today. And to make a parenthesis, we are using this one to measure offspring in reproduction assays. This model has a filter in which the nematodes are placed on one side only, and the M9 buffer is spaced and the offsprings are collected. Here you can see this design of a microfluidic device for reproduction studies. It has 3 rows of pillars, and the separation between these pillars is 30 micrometers, and the distance between the rows is 100 micrometers. Well, following with chemotaxis, the second objective was to determine a methodology for chemotaxis analysis. The design consists of an intermediate curved channel in which the nematodes are placed. Besides, it has 2 separate straight side channels, one of them for the control and the other one for the attractant. Therefore, the device is divided in 2 areas, control area and the attractant area. And now, which is the procedure? First, the nematodes, in this case N2, the strain N2, are placed inside the microfluidic device, and they are distributed along the curved channels. We use it between 40 and 60 nematodes. Then the buffer or medium of control and the attractant are placed in the linear channels. And finally, the nematodes are counted on each area of the device at different times. Here you can see a video with the device loaded with nematodes and test components. Okay. You can see the movement of the nematodes along the device. Okay. At this point, 2 questions were established. First, which compound causes the strongest attractions inside the microfluidic device? For this purpose, the following tests were carried out. E. coli confronts with buffer M9 and LB medium, an exudate of choline, isoamyl alcohol, sodium chloride, and 2-butanone were also tested. As you can see in this image, the attractant with the highest percentage of nematodes was selected. In this case, it was 2-butanone. At the same time, the second question was, at what point is the highest chemotaxis index obtained? For this purpose, the nematodes were counted at different points in time for 3 hours. In the left side, you can see the photos of the movement of the nematodes at different times. The red arrows show how they move to the attracting area. Then, with this data, the chemotaxis index was calculated using the following expression. Number of nematodes in the attracting area minus the number of nematodes in the control area divided by the total number of nematodes. As you can see in this image, the highest concentrations index occurs at 150 minutes and then begins to decrease slightly. It is for the reason that this time was selected as the minimum time for the following test. So the system consists in 2-butanone and a time of 150 minutes. Now, with these parameters determined in the device, we proceeded with the third objective, which is the applications of the device in toxicological tests. For this purpose, 4 compounds known to have possible neurotoxicological effects were used. These compounds are paraquat, glyphosate, cypermethrin, cialotrin, and 3 concentrations were performed for each one. L1 nematodes were treated for 72 hours. After this time, the assay concentrations was selected by 2 aspects. The nematodes must show growth and locomotion. Then the nematodes of the selected concentrations are washed with distilled water 3 times. They were started for 60 minutes before being placed in the device to finally evaluate their chemotaxis index. And these were the results obtained. Here you can see the chemotaxis index at 150 minutes for the control nematodes, and the toxicant treated nematodes. As you can see, in relation to the control, the treated nematodes have a lower chemotaxis index, being more evident in the treated nematodes with glyphosate and cialotrin, which also have greater dispersion. Here is the decreasing list of the chemotaxis index according to the toxic applied, with glyphosate being the compound that most affects the chemotaxis behavior. In conclusion, Microfluidic devices were developed in PDMS, which implies design, fabrication, and applications in toxicological studies. The methodology for the applications of the microfluidic system in these studies was determined through the applications of different attractants at different times, being selected to butanol at 150 minutes. And finally, the microfluidic device was applied to determine the chemotaxis behavior with nematodes previously treated with toxicants. Showing that they were affected in their chemotaxis behavior, being the treatment with glyphosate, the one that most affected the nematode.